The in vitro fertilization, or IVF, is a scientific process wherein the fertilization of the egg cell occurs outside the woman's body. Specifically, it is the fertilization of an egg in a laboratory dish or test tube. The first known success of an IVF was the birth of the test tube baby, Louis Brown, in 1978. Since then, there have been lots of changes and improvements in the procedure of the IVF. In 1978, when the first test tube baby was conceived, a single pre-ovulatory oocyte from Brown's mother was taken and fertilized, which then resulted to an 8-cell embryo that was transferred back to the mother's uterus. However, modern-day in vitro fertilization is much more complicated, but has a higher chance of success. First is ovarian stimulation, wherein synthetic hormones will be used to stimulate the mother's ovaries in order to produce multiple eggs. It usually takes 1 to 2 weeks in order to prepare the egg cells for retrieval. Several medications are used during this stage of the in vitro fertilization, such as 1. Follicle-stimulating hormone, a luteinizing hormone, or a combination of both for the development of more than one egg at a time. Second, human chorionic gonadotropin for oocyte maturation. And lastly, medications that would prevent premature ovulation, as well as progesterone supplements to prepare the uterus lining, are given on the day of egg retrieval or at the time of embryo transfer. Vaginal ultrasound and blood tests are also conducted to monitor follicle development and monitor the mother's response to the medications. In egg retrieval, the process can be done 34 to 36 weeks after the final injection and before ovulation. First, the mother will be sedated and given pain medication, then the egg retrieval process will begin. The usual retrieval method is referred to as the transvaginal ultrasound aspiration. In this process, an ultrasound probe will be used in order to identify the follicles needed. It will be inserted through the vagina, and a thin needle will be inserted next through an ultrasound guide to retrieve the eggs in the follicles. In sperm preparation, there are 3 ways to prepare the sperm to be used in the in vitro fertilization. 1. Using the sperm of the mother's partner, where the partner will provide a semen sample on the morning of the egg retrieval. Second is testicular aspiration, where the sperm is directly extracted from the testicle with the use of needle or surgical procedure. Lastly, the use of donor sperm. In the laboratory, the sperm will then be separated from the semen fluid. In fertilization and embryo culture, there are 2 methods commonly conducted to attempt fertilization. 1. Conventional insemination, where the mature eggs and healthy sperm are mixed in a petri dish. It would then be incubated after. Second is intracytoplasmic sperm injection, where a single healthy sperm is injected directly into each mature egg. This process is often conducted either if the person has a history of failed IVF cycle or if there is a problem in the semen quality or number. In embryo transfer, it usually occurs 2 to 5 days after egg retrieval. A mild sedative might be given to the mother due to the possibility of experiencing mild cramping. A syringe attached at the end of a catheter will be inserted into the vagina to the uterus. It contains 1 or more fertilized embryos that are suspended in a small amount of fluid. The syringe is used to place the embryos in the uterus. Approximately 6 to 10 days after the egg retrieval, if the transfer is successful, an embryo will embed in the uterus lining. And lastly, the pregnancy stage. Approximately 12 to 14 days or 2 weeks after the egg retrieval, the specialist will determine if the IVF is successful. Using blood samples, the doctor will determine if the mother is pregnant or not. The excess embryos are either discarded or frozen through cryopreservation. Cryopreservation is the process of preserving biological constructs by cooling the samples to very low temperatures. The in vitro fertilization can help infertile couples to probably conceive a child. With the use of technology, the impossible in the natural reproductive process can be turned into a possible through IVF. However, IVF can also cause negative effects and risk to the mother. According to Kilbaya, the most serious effect could be multiple pregnancies and ovarian hyperstimulation syndrome. There can also be other effects such as ovarian torsion, ectopic pregnancy, anxiety, depression, and others. IVF is also used in animals, which results in epigenetic changes, affecting various gene expressions, specifically imprinted genes that play a significant role in normal animal development. It also decreases and conserves biodiversity. For instance, excessive use of the same sperm decreases genetic diversity, and recessive characteristics can cause genetic diseases. Assistive reproductive technology or ART, such as IVF, can help with the conservation of rare and endangered species. Because of this, it sparked discourses on the technology's ethical concerns. First, embryos in the uterus may be destroyed in the process of selective pregnancy. If the embryo is perceived as a human being with rights, arresting its development will be considered unethical or an act of murder. Also, medical interventions may produce harm to the infertile couple. The success of IVF depends on the number of embryos transferred to the uterus. Because of the low survival rate of embryos in IVF, the more transfers made, the greater the chance of pregnancy. However, this also increases the chances of multiple pregnancy, which presents a threat to the physical and mental health of the mother. Studies also show that children born of IVF have a significantly greater risk for spina bifida and transposition of the great vessels. Some of the drugs administered to women to stimulate the production of eggs also increase the risk of serious birth defects. Given these evidences, some contend that IVF has an obligation to prove that their processes are safe and that it should not be used until proven safe. According to Robertson, these observations do not justify banning the technique, in that without these techniques, the children would not have been born at all. We are left with the question, is being alive better than not existing? Does the benefit of existence outweigh the harm of birth defects? With the conservatism of society, IVF remains a debatable issue despite its popularity in science. For instance, the Church deeply condemns the idea of abortion, art, and other biotechnology that disrupts nature. Because of the inability to separate the traditional views of the Church from lawmaking, IVF remains inaccessible to the middle to lower class due to a lack of health assistance. IVF also involves a procedure that discards excess human embryos. As the Philippine Constitution understands that life starts at conception, this practice is considered abortion and violates the unborn baby's human rights. Religious groups express their dissent on the growth of IVF centers in the country. They argue that such practices have implications for the sanctity of human life and the meaning of marriage. Although the Philippines have been practicing IVF for years, the country does not have a legal framework for motherhood yet. The conventional and legal idea of motherhood is conceiving, bearing, and delivering a child. Under this, as regarded by the Civil Register Act, the surrogate mother would be the registrable mother. To address these issues, we formulated a few recommendations. 1. Discarding excess human embryos can be avoided by creating only the required number of embryos needed, which can be safely implanted in the mother's uterus at the time they are produced. Cryopreservation should also be avoided if there is no certainty in conducting another IVF in the future. Some are also concerned about the consequences of surrogacy to the meaning of marriage. Christians argue that IVF practices can overstep boundaries as it creates artificial divisions between sex, conception, and childbirth. The meaning of marriage can be preserved by making IVF use exclusive only to married couples with no third-party involvement. No sperm donors, no egg donors, and no surrogacy. However, this should be implemented to the Christian couples only, in consideration to the non-Christian infertile couples who wish to try conceiving children through IVF. Lastly, to address the issue regarding the framework of motherhood, it would be beneficial for the families who would choose to go under IVF to have an assurance by the law that the child would be registered under the name of the expectant mother and not the surrogate.